Here we are producing the electricity. So you may also produce some fuel that is called as the gobar gas. Gas is helpful in the production of heat, and the microbes may also produce the electron. The flow of electron is called as electricity, you know. Yes. So here we are going to produce electricity by using the bacterial cell. Yes. So the bacterial cell setup, microbial fuel cell setup, has the substrate to which you are going to add the bacteria. So this bacteria will digest the organic material. Yes, digest the organic material and produce the proton and the electrons. The reaction is called as the redox reaction. That is, it involves oxidation and also the reduction reaction. So once the electron is being produced, it will just flow from anode to cathode. So the flow of electron produces the electricity. You can see the bulb glowing. The proton which is being produced will move to the cathode through the proton exchange membrane. Now the membrane which is present in the middle is called as the proton exchange membrane. So this proton will get added to oxygen to form the water. Okay, so that is for the microbial fuel cell. Then coming to gobar gas or biogas. What is gobar gas or biogas? The name tells you biogas. The gas is produced from the biological matter, okay, or organic matter. So mixture of different gases by anaerobic digestion of organic material. Yes, what is this composed of? Mainly composed of the methane and also the hydrogen and carbon dioxide. Major part of the gobar gas or the biogas consists of methane. So name itself tells you the methane is produced by the bacteria. Okay, they are called as methanogens. What are they called? Methanogens, methane producing agents, or methanogens or methanobacteria. So for that we need the organic matter. Organic matter or the municipal waste, that is agricultural waste, plant waste, household waste, whatever be the waste that can be allowed to undergo the anaerobic digestion by the bacteria, that is methanobacterium, to produce the gas. Yes, so once the gas is produced, what is the nature of that particular gas? It is a devoid. Devoid means no, it is not present. Devoid of smell, and it will not produce smoke, and it burns with the blue flame. That is the nature of this gobar gas. Yes, so the commercial production of the gobar gas is done in the biogas plant. Biogas plant. So there is a biogas plant Yes, in that way the weeds are harmful for the crops. Those pests, insect, weeds and herbs should be controlled with the help of the living organism, because if you use a chemical material like chemical insecticide, pesticide, weedicide, it will spoil the quality of the soil, because they are all chemical material makes the soil more and more dry. It cannot withstand the dryness, or it cannot hold the water in them. Yes, so then we are going for the life is controlled by life. Next one, biopesticide. 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 Here we have the ladybird beetle. This will eat on the aphid, which is going to affect the crop. The dragonflies can eat the mosquito larvae, so that the population of the mosquito will be reduced. Then coming to Bacillus thuringiensis. Bacillus thuringiensis is very, very important. So this is a bacteria. Bacillus thuringiensis is a bacteria. As you know, bacteria in an inactive state, they form the spores. Spores are like the seeds. When they come out to the suitable substratum, it will start growing. Yes, so we have to take Bacillus thuringiensis spores and you give it for the plant. You apply it or spray it over the plant. These spores will be ingested by the insects present in the crop. Once it is getting ingested, this spore will have the toxin that is called as delta endotoxin. It is in the form of insoluble crystal. Insoluble crystal. What is it? Delta endotoxin. Once it is getting ingested by the insect, in the intestine of the insect, it undergoes chemical reaction. Due to the alkaline nature of the insect gut, it is getting converted into soluble form. Once it is getting dissolved in the gut, it will just go into the layers of the gut and it paralyzes the digestive system. Paralyzed means it will not be moving anymore. Once the food is being taken, the digestive system has to move. Then only the further proceeding of the movement of the food occurs. So this toxin will stop the movement in the gut, so that the insect will not ingest further. It will die without eating. That is the major aim for the using Bacillus thuringiensis spores. Bacillus thuringiensis produces endotoxin, as you know, that is coded by the gene. That gene is called Cry gene. CRY gene. Yes, Cry gene produces endotoxin. That is found by the scientists. Then they have taken the Cry gene and they have infused into the plant cell when it is in the condition of the embryo. So in the seed stage itself, the gene is infused to make genetically modified plant. That plant will be resistant to the insect, because the cell has got the gene, that is Cry gene, that will automatically produce the endotoxin, so that the plant becomes resistant to the insect. One such variety of crop is produced that is called as the BT cotton. We shall continue with the bioherbicides. What are bioherbicides? These are the species microbes which will not allow the other plants to grow along with the crops. So Phytophthora alvivora controls the weeds in the citrus crop. When you make the citrus crop to grow in your field, you can make use of this organism which will avoid the growth of other plants. Then Trichoderma, that controls Trichoderma species, controls the pathogen cannot take the phosphorus from the soil, and the fungi will automatically get the nourishment from the plants too. Then this fungi makes the plant become soil salinity resistant and also the drought resistant. During the dry condition also plant will be growing without any damage or any dryness because of the presence of this fungi, and also it becomes resistant to salinity. In the water which is salty also the plant is able to grow with the help of this fungi. Then coming to cyanobacteria. Cyanobacteria, when you put it in the paddy field, they help in the growth of the plant by producing the growth promoting substances. So the cyanobacteria produce a growth promoting substance. They are indole 3 acetic acid, indole 3 butyric acid, naphthalene acetic acid, and some of the other amino acids, proteins and vitamins are also produced by the cyanobacteria. In that way it acts as a biofertilizer. Then coming to organic farming. What is organic farming? It is cultivation of plants and animals without the use of the synthetic material. Yes, so plants and animals are grown in a natural way without synthetic material. What is the advantage of this? It minimizes the pollution level. When we use a chemical, it pollutes the soil. In turn, the water that is getting minimized. So microbes are used as the fertilizers, pesticides, insecticides, herbicides, weedicides. As we have seen all the examples there, they are all used in the organic farming. Then we move on for the very important topic, that is bioremediation. What is bioremediation? Microbes used to degrade the pollutant. What is pollutant? The agent that causes pollution, that you call it as pollutant. That pollutant can be degraded or digested or made inactive in the presence of certain microbes. That is what we call it as bioremediation. Yes, bioremediation can be done by the anaerobic bacteria and also aerobic bacteria. First three examples are aerobic bacteria. First one is Pseudomonas butida. So what is Pseudomonas butida? It is genetically modified organism which will digest the hydrocarbons. It is present in the oil. When there is an oil spill, you can apply Pseudomonas butida to that particular area. It will digest the oil spill and make the area pollutant free. This is created by Anand Mohan Chakrabarty. He is an American scientist. He is living in America, but he was born in India. Then Nitrosomonas euphoria, that is the bacteria which is able to digest benzene. And Ideonella sapiensis is very important. This species of bacteria can produce the enzyme which will digest PET plastics. As you know, the plastic pollution is a major threat to the world now. These bacteria can digest the plastic and minimize the pollution. Once these PET plastics are getting digested, it produces tetraphthalic acid and ethylene. Now we will start with anaerobic bacteria, which is helpful in bioremediation. Bioremediation is to reduce or degrade the pollutant with the help of anaerobic bacteria. We have few of the species which helps in the digestion of the material which is polluting the environment. Here, Dechloromonas aromatica is a bacteria which is able to degrade benzene to toluene and xylene. Then Phanerochaetum cryosporium is a bacteria which is able to digest the polyaromatic hydrocarbons, dye, cyanides, etc. Then Dihalocoides is a bacteria which is able to digest trichloroethane to ethane. Trichloroethane is digested to ethane. Then Phacelotheopus microspora is a species which is able to digest polyurethane. Is a material which is formed after the digestion and it is not harmful also. Now you may write all the key points which are written on the board, and also book back questions. 8th lesson is over for you. 8th lesson book back question you can write in your classwork and you can send it on Wednesday. Thank you.